full virus genomes that are in the databases, 70% of them now are this variant. This variant has gone, as we say, to fixation. So it's pretty much the only variant that people are sequencing, and that's all over the planet. The variant has clearly dominated the outbreak.
It just happens to be the one that's landed really well wherever it spread. But that's what you're looking at, is whether actually there are mechanistic reasons why it's doing better.
Right. So we're focusing on what effects this variant would have on the structure of the protein that it encodes, what effects those changes in structure might have on the function of the virus, and whether we can at least generate data that supports the idea that this variant somehow has an advantage at the molecular level that we can understand that would be consistent, at least, with this virus taking over because it has an advantage.
And that's because this protein is on the surface. It's the key that unlocks its way to get into the cells of our lungs and so on.
So this virus is the biggest of the RNA viruses. It's very complex. It has many, many proteins that it makes. There's mainly one protein on the surface, the barium particle with the new target cell. So without this machine working properly, without it being tuned really precisely, the virus will not be infectious, or it will not be transmissible.
So in your lab, you're able to work with this protein. How do you study it?
We put some limiting amount of the virus onto a target cell that it makes. See, there's pound for pound, mainly one protein on the surface, barium particle with the new target cell.
And the answer?
The answer is that machine working properly without it being tuned really precisely, better able to the virus will not be infectious,
3 or 10.
or
this is
it
not
will
a small
not be
effect,
transmissible.
Then. 3 or 10. So in your lab, you're able to work with this protein. How do you study it?
An incredibly complicated limiting amount of the virus.
It has
to incorporate itself into a virus. Mainly one protein on the surface, stable particle with such target cell. That virus. And the answer is this. Through the virus working properly without it being tuned really precisely, the virus will not be infectious.
3 or 10. This is not a small effect. 3 or 10. So in your lab, you're able to work with this protein. How do you study it?
Incredibly complicated limiting amount of the virus. It has to incorporate itself into the virus. Mainly one protein on the surface, stable particle with such target cell. That virus. And the answer is this. Through the virus somewhere properly without it being tuned, should make drugs better able to use the virus will not be infectious. So much of it will not be changed to target cells.
So in your lab, you're able to work with this protein. How do you study it?
Incredibly complicated limiting amount of the virus. It has to incorporate itself into the virus. Mainly one protein on the surface, stable particle with such that virus. And the answer is this. Through the virus properly without it being should make drugs better able to use the virus will not be infectious. So much of it will not be changed to target cells.
So in your lab, you're able to work with this protein. How do you study it?
Incredibly complicated measure of the virus. It has to incorporate itself into the virus. Mainly one protein on the surface, stable particle with such that virus. And the answer is exactly at that point. Should make drugs better able to use the virus will not be infectious. So much of it will not be changed to target cells.
So in your lab, you're able to work with
this protein
peoples, my sample will go into, let's say, 3 test tubes. Call them test tube number 1, number 3, and number 5. Number 3 and number 4. It's like a barcode, so if my sample will go into, let's say, 3 test tubes, I am carrying the virus, test tube number 1, number 3, and number 5. Number 3 and number 4. It's like a barcode, so if my sample will go into, let's say, 3 test tubes, I am carrying the virus, test tube number 1, number 3, and number 5. It's like a barcode, so if my sample will go into, let's say, 3 test tubes, I am carrying the virus, test tube number 1, number 3, and number 5. It's like a barcode, so if my sample will go into, let's say, 3 test tubes, I am carrying the virus, test tube number 1, number 3, and number 5. It's like a barcode, so if my sample will go into, let's say, 3 test tubes, I am carrying the virus, test tube number 1, number 3, and number 5. Leon, you're the one actually has to do barcodes, but you have my trying to go into, let's say, 3 test tubes, I am carrying the virus, test tube number 1, number 3, and number 5. Leon, you're the one actually has to do barcodes, but you have my trying to go into, let's say, 3 test tubes, I am carrying the virus, test tube number 1, number 3, and number 5. Leon, you're the one actually has to do barcodes, but you have my trying to go into, let's say, 3 test tubes, I am carrying the virus, test tube number 1, number 3, and number 5.
And among these, half of them have been done by pulling up.
Leon, you're the one actually has to do barcodes, but you have my trying to go into, let's say, 3 test tubes, I am carrying the virus, test tube number 1, number 3, and number 5.
When you do the pulling, half of them can go up to 7.
Leon, you're the one actually has to do barcodes, but you have my trying to go into, let's say, 3 test tubes, I am carrying the virus, test tube number 1, number 3, and number 5.
Yes, the pulling approach is applied when you have a low
because I guess if there was only one infected person in 100, the very people, you need to test 100 people, that'd be 100 tests just to find one successful identification. This is much more efficient. Because I guess if there was only one infected person in 100, the very
Then we be testing on 5 pools. If you have one pool which is positive, so you go back and then you test one by one. But meantime already we're done with the other pool.
This is a very practical use of the skills that you sort of assembled at the Affinity for Mathematical Sciences.
So you go back and test one by one.
Particularly exciting is to see the most sophisticated.
Which are now interested, South Africa, for example, to see if they can implement in their country. So there are many countries now, especially on the African continent, being tested. It's not only for developing countries like in Africa. So other countries also can South Africa, for example. I can give you an example when you want just to lift the lockdown, or if you have to test, for example, in the there are many countries also other, especially like African continent, being tested. It's not only for developing countries like in Africa. In any case, other countries also can South Africa, for example. I can give you an example when you want just to lift the lockdown, or if you have to test, for example, in the there are many countries also other.
I'm sure you would agree with that, Wilfred. I mean, in the UK, I know the system, people don't think is keeping up with the number of tests they need to do. In America, we clearly see to be more efficient and to be quicker seems to be very important. Using a approach like this.
Applying this approach, not that it cannot be applied, but the efficiency gain becomes less.
Neil, you're in the UK at the moment. Are you using your position as a esteemed mathematician, esteemed scientist, to go around and knock on doors?
Not there in the UK at the moment. I think this is rewarding the government of Rwanda in particular, who have been very determined to control the prevalence, control the virus. It's just great to see them succeed.
Neil,
I do think it'll take a little time for the UK to realise it's behind the curve, and other countries. But yes, we think once the benefits of the system are appreciated, I think, as Wilfred says, this is rewarding the government of Rwanda in particular. The great thing about it very incentivizes government to control the prevalence, control the virus, then it's just great to see them succeed.
Neil, you're in the UK at the moment. Are you using your position as a esteemed mathematician?
Yes, we think once they're in the UK system appreciated, I think this is rewarding the government of Rwanda in particular to assure you about it very determined government to control the prevalence, control the virus, and all just great to see them succeed.
Neil, you're in the UK at the moment. Are you using your position as a esteemed mathematician?
Yes, we think once they're in the UK system appreciated, I think this is rewarding the government of Rwanda in particular to assure you about it very determined government to control the prevalence, control the virus, and all just great to see them succeed.
Neil, you're in the UK at the moment. Are you using your position as a esteemed mathematician?
Yes, we think once they're in the UK system appreciated, I think this is rewarding the government of Rwanda in particular to assure you about it very determined. The problem of uniquely identifying infected individuals, as Wilfred said, the higher dimensions are actually an enormous advance.
Thanks to Neil Tierrock, Leon Matiza and Wilfred Ndifon. Let's put coronavirus to one side and return yet again to climate change. It's only a couple of weeks since we were talking about the scorching temperatures in Siberia. 6 months earlier, it was the opposite end of the planet, in Australia, that they were suffering a record-breaking heatwave. You'll remember the extraordinary wildfires there. Well, at the University of New South Wales, climate scientist Sarah Perkins Kirkpatrick